Welcome to today's Explainer, where we're going to completely decode the intricate molecular blueprints of life. We'll be starting from the absolute most basic chemical building blocks and watching them assemble into fully functional, incredibly complex biological machines. It's going to be a fun ride. Here's our game plan for today. We're going to take a builder's journey together through 5 key areas. We'll start with phospholipids and cell walls, look at how to test for hidden lipids, dive into the building blocks known as amino acids, explore the absolute molecular origami of protein folding, and wrap up with a showdown: globular versus fibrous proteins. Section 1, phospholipids, the cell walls. How cells build their barriers. Okay, let's just dive right into this and look at how our bodies pour the foundation. Every single cell in your body needs a barrier, right? Well, we build these fundamental walls using a very specific, highly specialized type of lipid. To understand how they work, you have to picture the architectural blueprint of this barrier, which is the phospholipid molecule. It has a very distinct shape. Up top, you've got a polar phosphate group. That's the head. This head is attached to a glycerol backbone, which then anchors 2 long fatty acid carbon tails extending downwards. What's literally the coolest thing about this molecule is its split personality. Think of them as microscopic molecular bouncers. The hydrophilic heads absolutely love water. They are totally soluble and polar. But the hydrophobic fatty acid tails, they are entirely repelled by water. So when they arrange themselves in a double layer to form a cell wall, the heads face outward toward the watery environments inside and outside the cell, while the tails hide safely inside the layer. It creates a highly effective, impermeable wall that keeps unwanted water-soluble substances out. But lipids are incredibly versatile. They do way more than just build biological walls. They actually serve as massive energy reserves because of the huge number of carbon-hydrogen bonds they pack in. Our bodies store them heavily under our skin and around vital organs like the kidneys, acting as essential thermal insulators to stop heat loss. And amazingly, when they're oxidized during respiration, they even act as a metabolic source of water. Section 2, testing for hidden lipids, the emulsion test. Now, moving along, let's say you were in a lab and wanted to figure out if fats were hiding in some mysterious substance. How exactly do you go about it? Well, the classic emulsion test is elegantly simple. It's basically a 3-step recipe. Step 1, take your substance and shake it thoroughly with ethanol, which dissolves any lipids that might be hanging out in there. Step 2, pour that ethanol mixture directly into some water. If lipids were actually in that original substance, step 3 happens automatically. A cloudy white suspension will immediately form in the water, giving you a rock-solid positive result. Section 3, amino acids, building blocks, manufacturing the steel beams of life. All right, so our cellular foundation is poured and our lipid walls are up. Now it's time to manufacture the steel beams of our biological skyscraper, proteins. And to understand proteins, we really need to look at what they're made of. Every single protein in existence starts with the exact same basic structural unit, an amino acid. It contains an amino group, which is the NH2, an acid group, the COOH, and sitting right there on top is the crucial R group. Now, this R group is the wild card. It changes from molecule to molecule, and it's what gives each specific amino acid its totally unique chemical identity. Now, here's where it gets interesting. When these individual amino acids link together to form a chain, they don't just snap together like magnets. They go through what we call a condensation reaction. During this process, they literally lose a water molecule in order to form a super strong peptide bond, creating a continuous chain known as a polypeptide. And these polypeptide chains, they're the ultimate multi-tools of the body. Once fully formed, they take on an astounding variety of roles. They become the enzymes that drive our metabolism, the hormones that send signals, the hemoglobin carrying our oxygen, our defensive antibodies, and even the tough structural collagen, actin, and myosin that literally make up our hair, our nails, and our muscle fibers. Section 4, molecular origami, protein folding, turning chains into 3D machines. But here's the catch. A straight floppy chain of amino acids isn't going to do all that heavy lifting for you. To actually function, it has to be folded into a highly precise shape, much like complex 3D molecular origami. This fascinating folding journey happens in 4 distinct, beautifully choreographed stages. It builds all the way up from a simple linear sequence right into a complex, multi-chain structural marvel. It all begins with the primary structure. This is simply the specific, linear sequence of amino acids in the polypeptide chain. If you trace the ends of this chain, you'll find an amino terminal, known as the N end on one side, and a carboxyl terminal, or C end, on the very opposite side. Now, as we move forward to the secondary structure, hydrogen bonding along the backbone forces the chain to fold. It naturally wants to twist into a tight, regular spiral called an alpha helix, or it might fold back and forth on itself to create a looser, flatter structure known as a beta-pleated sheet. But our molecular origami doesn't stop there. To get to that compact, highly functional 3D shape known as the tertiary structure, the molecule relies on a wildly complex internal network of bonds. These bonds connect those diverse wildcard R groups we talked about earlier, pulling the coils and sheets into a tight, three-dimensional tangle. When we break down the specific bonds holding this 3D shape together, you've got strong covalent disulfide bonds forming between sulfur-containing cysteine molecules, working right alongside weaker ionic bonds, hydrogen bonds, and hydrophobic interactions. But pay attention to what breaks them. High temperatures and shifts in pH can easily snap these specific bonds, unraveling the protein entirely. A perfect real-world example of this compact tertiary structure in action is a little something called myoglobin. This is a single, intricately folded polypeptide chain that stores oxygen directly in our muscle tissue. Fun fact for you, it's also the exact molecule that gives raw meat its distinct red color. Finally, we reach the quaternary structure. This is the absolute peak of the origami process. This is where multiple separate polypeptide chains, and sometimes even non-protein components like a heme group, combine together to form a massive, fully functional megamolecule. Section 5. Globular versus fibrous proteins. Shape equals purpose. This brings us to the ultimate showdown, and really the central driving theme of our whole explainer today: how a molecule's final folded shape perfectly dictates its ultimate job in the human body. The contrast between the two main types of proteins is absolutely striking. On one hand, you have globular proteins. They are folded into neat spherical shapes, dissolve easily in water, and act as highly active metabolic workers. On the flip side, you have fibrous proteins. These guys form long strands, remain stubbornly insoluble, and their job is entirely structural. Now, how do globular proteins manage their incredible solubility trick? It's all because of a very specific folding pattern. Their water-fearing, hydrophobic R groups hide safely in the center of the sphere, completely away from the fluid environment. Meanwhile, their water-loving, hydrophilic R groups are pushed to the outside edge. This allows water molecules to just cluster around them and easily dissolve the protein. Fibrous proteins, on the other hand, are the complete opposite. They do not fold into neat, soluble little spheres, because they don't need to dissolve, and they don't need to be metabolically active. They just need to be tough, inflexible, structural cables that hold the actual physical architecture of your body together. Which leaves us with a fascinating and maybe slightly terrifying thought to end our explainer today. We've seen how perfectly these lipid walls are built and how precisely these protein chains have to fold into their active shapes. But if just a slight shift in your body's temperature or a tiny change in its pH can unravel these bonds and destroy the shape of these vital proteins, just how delicate is the invisible architectural blueprint holding you together right now?